Hello everyone, welcome to our presentation. The topic of our presentation is HPLC, High Performance Liquid Chromatography or High Pressure Liquid Chromatography. HPLC is a chromatographic technique used to separate a mixture of components in analytical chemistry and biochemistry with the purpose of identifying, quantifying or purifying the individual components of the mixture. The mobile phase of HPLC may be a single liquid or a mixture of liquids which is pumped into the column at high pressure of the order 400 to 1000 atmosphere and the flow rate is adjusted to 0.1 to 10 milliliter per minute. Advantages of HPLC: high speed of separation, excellent column separation, solvent consumption is minimum, sensitive and accurate, separation can be done at ambient temperature. Applications of HPLC. HPLC is probably the most universal type of analytical procedure. Its application area includes: first one, quality control. HPLC is widely used in quality control to perform several tasks including identification of unknown compounds in a mixture, verification of composition of mixtures, detection of degradation products of a substance over time. Coming to the next application of HPLC, that is environmental monitoring. It includes the analysis of water quality, air quality monitoring, soil analysis, and food and beverage analysis. Water quality analysis involves the testing for contaminants and pollutants in water supplies. In air quality monitoring, the volatile organic compounds and other pollutants in the air is tested. Then soil analysis. It is the testing for toxic metals, pesticides and other contaminants in soil. HPLC is commonly used for biomedical research for the analysis and purification of biological molecules such as proteins, peptides and small molecules. The second application of HPLC is process control. HPLC is used in process control to monitor and optimize chemical process. It can be used to monitor product purity and quality, identify and quantify impurities, ensure process consistency, improve process efficiency, monitor raw material quality.
In this animation, we are going to talk about instrumentation and working of HPLC. Full form of HPLC is High Performance Liquid Chromatography. HPLC instrument is constructed by combining different parts, which are solvents reservoir, which contains different solvents, HPLC pump degusser, which is attached with vacuum pump, solvent mixing valve, sample injector. Next part is pre-column, which is also known as guard column, and length of this column is 2 to 10 centimeters. Internal diameter is 4 to 5 millimeter, usually 4.6 millimeter. After that column, next one is analytical column, and length of this analytical column is 10 to 30 centimeters. Again, internal diameter of this analytical column is 4 to 5 millimeters, usually 4.6 millimeter. The analytical columns and pre-columns are made up of stainless steel and filled with a stationary phase. Commonly used stationary phase is octadecyl silica gel, also known as ODS silica gel, and common names of these columns are C18 columns. Next part is detector, and finally coming to the recorder. Now let us discuss the working of this HPLC instrument. First we will start the HPLC instrument. Then all parameters will be entered by the help of available HPLC software. And these parameters are solvent ratio, flow rate, runtime, type of elution, etc. After creating the method, solvent of the mobile phase will be pumped by the HPLC pump from the solvent reservoir towards the degusser. Now these solvents will come to the degusser, and degusser is attached with a vacuum pump which will create high vacuum. Therefore, any gas or air dissolved in the solvent will be removed online. because this may create a problem during elution. After degassing, solvents of the mobile phase will move towards the solvent mixing valve, and mixing of the solvents take place, which will help to produce uniform mobile phase. Now this mobile phase will run towards the pre-column or guard column. This guard column will remove any kind of impurity present in the mobile phase and protects analytical column from the impurities. In the next step, mobile phase will further move towards the analytical column, then finally goes to detector. In this condition, blank mobile phase will run for 10 minutes at least, which gives proper conditioning of the HPLC instrument. Now we will inject the required amount of sample through sample injector by the help of a micro syringe. After injecting the sample, it will pass through the guard column. Then it will enter into the analytical column, which contains stationary phase. Due to the difference in the affinity with stationary phase, compounds present in the sample will be separated inside the column and reaches to the detector and gives signals in the detector. Compounds having least affinity with the stationary phase will reach to the detector first, which is indicated by blue color. Then moderate affinity compound will reach to the detector, which is indicated by red color. And finally, highest affinity compound will reach to the detector at the end, which is indicated by green color. Ultimately, all signals will appear in the recorder. We have discussed the details about instrumentation and working of HPLC.